Hi, it's a pleasure to introduce Nadia Rosenthal, who is scientific director of the Bar Harbor Jackson Lab and is visiting for the day and is going to tell us about mice and translating human genetic variation to function.
Thank you. Great. Well, thank you for coming. Thanks to all of you online. Hope I keep you online. I hope you don't knock off because it's boring. I will not make it boring, I promise you. All right. So I'm going to start with just a little bit of history, because I know that George is a bit of a history buff. And you probably have never heard of the Jackson Laboratory. If you have, you've thought of just what place it sells mice. But actually, it's got quite an interesting history. And it all started with a woman named Abby Lathrop, who was a mouse fancier, and she was also quite a talented geneticist. She was interested in heritability. I mean, we're talking about, the 1900s here. So she had a number of adventures with dogs and then with guinea pigs. You can see her observing one of her pets there. But eventually realized she could fit more mice in her big barn in Granby, Massachusetts, than guinea pigs. And she ended up with about 11,000 of them. And she kept meticulous notes about when these mice were born and died and of what, and ended up being sought after by a number of notable people at Harvard. In particular, a guy named Castle, who started to get excited about the mice that Abby had so carefully annotated. A lot of these were fancy mice. That's what people had back then. And he was buying them by the pound, which always cracks me up. I've always tried to figure out how many mice would fit in them. Anyway, so he had two rather remarkable students. One was C.C. Little, who was quite a character. You can see him there lighting his pipe. He was a bit of an Errol Flynn. And he actually came up with an incredibly prescient idea that cancer might be genetic. And if you think about it, early 1900s, that wouldn't have been what everybody thought about. There was a quieter fellow named George Snell who was studying genetic linkage with Abby's mice. In any case, just fast forward, C.C. Little became the president of the University of Maine very early in his life, and he built the laboratory, the original laboratory in Bar Harbor. Theoretically, it was because you could more easily heat mice than air condition them, and Maine is known as the air-conditioned state. But actually, it was because he was entertaining all the wives of the wealthy summer people who would go up and build summer cottages with 40 bathrooms, and then go back to Philadelphia for the week, and the wives would spend their money by supporting C.C. Little, who would convince them that this was the answer to their ills. And probably the most gullible was the wife of Roscoe B. Jackson, because somehow he managed to give a gift. I wonder if it was him or his wife that wrote that check, which actually established the Jackson Laboratory. And for those of you who know anything about mice, it's rather extraordinary that we're still looking at a mouse called C57 Black 6, which was the 57th mouse from Abby's barn of that particular color. And there you have it. The rest is history. So in any case, there was that other quiet gentleman. You don't hear much about him until later, because he took years and years using congenic strains to discover the major histocompatibility locus at JAX and won the Nobel Prize for that. So now, if we just fast forward, this is where it is. It's in the middle of Acadia National Park with the Atlantic Ocean beyond, and there's about 3,000 people altogether on the various campuses. This is just the Bar Harbor campus, which is headquarters. 2 million mice on the shelf, serves about 2,400 organizations, 68 countries, all getting mice from the JAX, maintains 15,000 different strains. And the question is, why are we doing this ridiculous thing? So I'm going to try to spend the rest of my talk explaining to you why we have 15,000 mice and why it's important. And it's very clear to an epidemiological group like yourself that people are not all created equal. And as a result, that means that the diseases that they get are also not created equal. So if you think about it, that then pertains to the drugs we give people. So here are the rather disappointing number of failures that you see with any given drug in these various categories of illness. And what that tells you is that, for instance, cancer drugs, 75% of the population actually can't benefit from these drugs because the drugs are not set up to be specific for their particular cancer. It's not something that the pharma companies want you to know about, but it's really one of the reasons why we have to think about the variation in the population. As I said, I'm preaching to the choir here, and this obviously involves networks of genes, not just genes in their isolation, and the distinct disturbances amongst the networks of these genes is what really causes most of our diseases, keeping out things like very rare diseases, which are monogenic. So for the last 100 years, we've been using these little guys as models for all these different diseases. And as we know, these are kept as inbred strains. They're homozygous across their entire genome. That means that any given mouse is identical to the next one. Reproducible phenotypes emerge from that. If you order a C57 Black 6 mouse from Jack's now, you will be ordering almost the same genetic mouse that you ordered 20 years ago, because we refresh each of these strains every 10 generations with repositories of the original strain frozen down in order to avoid genetic drift, which is something that makes Jack's a bit special. But the problem with these inbred mice is that they are inbred, for one thing, and secondly, they're all different. For instance, AJ gets a very typical lung adenoma. C3H has hepatomas. And here's the famous C57 Black 6, which I like to refer to as the mouse. So low pain threshold, alcoholic, neurotic, sight and hearing loss very early in life, low bone density. So, I mean, is that really the model we want to use for everyone? Maybe there's some people for whom that is exactly the right model, but I'm not going to discuss that further. Anyway, are inbred mice good models? And of course, we have this problem, which is that we're genetically diverse and the mice are not. So what we'd like to propose is that actually adding genetic diversity to your animal model is the only way to actually be able to look at diseases in a realistic context. So a very, very small digression into where these mice came from. Abby was collecting mice from all over the world. And indeed, the mice that now contribute to the majority of our lab inbred strains are a mixture of mice that came from either domesticus at the bottom, the blue mouses from Western Europe, and that's more or less the mice that came over on the Mayflower, and that's more or less the mice that got into Abby Lathrop's barn. But she also was excited about Castaneus and Musculus from Southeastern Asia and Russia, Molossinus, which is a Japanese mouse, and they were very beautiful and fancy. And those gave rise to the fancy mice with the long fuzzy hair, which the Victorian ladies like to have on their shoulder. And this went on to a classic inbred strain. So we have Domesticus, Molossinus, Musculus, Castaneus. These are all subspecies of Musculus. And of course, you'd say, well, that's great. They're all nicely mixed up. The point is, there are these horrendous bottlenecks, and the bottlenecks came from the fact that when you're actually trying to perpetuate a strain, what you really are, what's really important is how many babies they have. So fertility became an enormous exclusion criteria for those who didn't breed well. And the other thing is that no one likes to get bitten by mice, and specifically the ladies who had them on their shoulder. So way beyond or before there were these bottlenecks about fertility, there was a bottleneck for docility, i.e. thick as a brick. So that's why when you reach in for a C57 black mouse, it actually allows you to pick it up. There's no mouse worth its salt that comes from the wild, which would put up with this. Okay, so, and when it comes to diversity, there are many differences between these various subspecies, but unfortunately, most of Abby's mice, from whom most of the mice we have derived, are really quite similar, and therefore not really that good for a mouse collection of genetically diverse, or GeDi mice, as we like to call them. Now, the problem is, when Trump came in, the word diversity became totally impossible. So we had to come up with some other ideas. And these are some of the things that various audiences have suggested so we can keep the acronym GeDi which of course everyone sort of likes. So anyway, if anyone has any GI starting names, please give me some ideas. Okay, so very briefly, I'm going to describe some of the current mouse panels that have been developed over the last 2 or 3 decades at the Jackson Laboratory and elsewhere. The person at the JAX who is probably the most well known for this is Gary Churchill, with whom George is collaborating. I guess you all are collaborating, some of you anyway. And I'll just describe exactly what these mouse panels are. And then I'm going to tell you about some very new mouse panels, which I think are going to be even more interesting for epidemiological study. Okay, so the collaborative cross was started 25 years ago using 8 different parental strains as starting material. 5 of those are right in the Abbey Lathrop, all the same kind of genome. And 3 of them are coming in from different subspecies, including Castaneus. That's PWK, Castaneus, and WSB. The idea was to do a number of crosses between these to increase the recombination frequency and mix up the genomes. And eventually, after about 20, then take each one of these and start to inbreed them so that you had reproducibility, but reproducibility across a number of different lines. And those are known as the collaborative cross. The idea that Gary and others had at the time was that these animals were going to be fantastic because you're going to have thousands of different crosses. But what they forgot was that these mice had been selected for fertility. And when you start crossing them, they lose that fertility, especially if there are subspecies differences in terms of meiotic disjunction and other issues of incompatibility. So right now, to be honest, the dirty laundry is we have about 30 that are any good. That's still pretty interesting, however. And in order to get around that problem, Gary and his Gary's collaborators decided to go a different direction. So taking the same 8 mice that have been extensively characterized, T to T genomes and everything else you'd need to know about them, he started outbreeding them right away and never stopped outbreeding them. And this is known as the diversity outcross. And there you have a lot more diversity because of the fact that you're not trying to inbreed. Okay, so I'm going to give you just a few vignettes on how these mice are used. And I'll use my own lab just because I have the slides. And basically, there are a number of really beautiful studies that have come out, which I think approximate some of the results that people are seeing in human genetics better than the original use of a single mouse. In this case, I'm using heart attack because I study the heart. Just a little biology. One of the things we discovered about, 5 or 6 years ago was that fibroblasts in your body are quite different depending on where they sit. And that's not surprising because I think fibroblast is a sort of outdated word that doesn't really capture mesenchymal cells that really do different things for different tissues. And specifically, we discovered that each tissue has fibroblasts that are expressing unique sets of genes that are characteristic of that tissue. So liver fibroblasts express only liver cells, liver genes mostly. This is just a cytoscape analysis. Skin, heart, etc. The ones on the outside are genes. Each circle is a gene that is expressed only in that fibroblast and not in other fibroblasts, whereas this middle stuff is what everybody needs as a fibroblast. So with that in mind, we started to look to see whether we could pick up some differences. And one of the things we found were that the developmental genes for each tissue were retained in the fibroblasts, which was peculiar because a lot of these genes are not normally expressed. And particularly cardiac fibroblasts express some genes that are normally turned off in development in every other cell type in the heart. So this was how we got to do this experiment, because we were wondering what happens to all those genes and why are they expressed only in fibroblasts. And what we discovered in the process was that just as we looked at the inflammatory, proliferative, and maturation phase, of course, the cardiomyocytes in the heart never divide again. So that's why you have this mess at the end, which is this huge fibrotic infarct. And that's what does you in and you have heart attacks. So someone in my lab, Elvira Forte, got very interested in the fact that there was a very rapid shift in the way fibroblasts were expressing genes very early on after an injury. And this is true also if you injure an animal with an infection. It's classic that you have these injury responsive fibroblasts that then slowly morph into a number of other fibroblasts, ultimately producing fibrosis. And in the process, we started asking questions about whether this was true in every mouse. So in this case, you're doing a classical tying off of the descending artery to produce an infarct, which is a pretty brutal experimental protocol. And we're much better off now than we were back 3 years ago. But to suffice it to say that even if you use two different mice, the C57 black 6 mouse and 129, also one of Abby's mice, this guy proceeds to heart failure eventually, whereas this guy has a huge inflammatory response. The fibrotic activity is extremely rapid but faulty, and the mouse essentially dies from a heart bursting. And when we looked to see what was going on with single cell, we discovered that indeed these two mice had a different population of cells for that very early stage of injury responsive fibroblasts. And these were fibroblasts that were expressing all sorts of nasties, metallothioneins, neutrophils, pro-inflammatory, all these very, very responsive genes that are typical of an acute inflammatory response. But it was really different in this animal, different enough to mean that the mouse was dead 5 days later. And when we started looking at other mice, and these are, of course, the 8 founders plus 1 extra one for the collaborative cross, we found all sorts of different ways in which these mice got heart disease. Some of them were really good. Oddly enough, the NOD, which is actually an autoimmune disease mouse, it gets type 1 diabetes, but impervious. And of course, now we can dig in and ask from the point of view of the cells, what exactly is making that mouse so impervious to an infarct? Something that's hard to do in humans. They don't usually you take their heart out after they've had a heart attack. So we went out to look at a whole bunch of different collaborative cross strains. And this was heroic work that a postdoc of mine, Kachi Salimova, did when I was still running the Regenerative Medicine Institute in Melbourne. And what we did was to look at a number, 36 or 32 different collaborative strains, doing infarcts on each one and looking at that versus the sham. And what we found was an extraordinary variety of responses, which is why I'm standing here today telling you that GeDi mice are interesting. This was the experiment that convinced me. And we got everything from a mouse called BOM-GB, which was bomb-proof, literally. You could tie that thing off and it just went poof, I don't care. Ejection fraction was fine and it survived, down to poor old Tofu, who kicked the bucket rather regularly. And just to give you a feeling for how different these were, this is a survival curve of those 36 versus ejection fraction and left ventricular volume, which is heart failure, essentially. And this is just a small subset of the data to give you a sense of how variable these were. Now, they're variable enough that if you're lucky, you can start to map genes that are associated with, let's say, in this case, left ventricular dilation, which is heart failure. And we were lucky enough to get ourselves a QTL that was above significance and ended up looking at that particular area where the very resistant animals differed in a number of ways from the very susceptible animals, and ultimately discovered a gene within this particular QTL called myo18B. I'm, obviously compressing a ton of failed experiments to tell you this story. And this is a gene that's expressed in multiple cell types, including the fibroblastic cell types. And when we found that it indeed was expressed in the heart, which was a good thing, and that it was actually also very important in sarcomeric function, we thought we'd have a better look. And what we discovered was that indeed in people, diseases of the heart, in particular Nemmeline myopathy with cardiomyopathy, involved myo-18b. Here's another one in a Swiss patient. My friend Damien Smedley, who used to work in Genomics England, found that patients at risk for heart disease carried a lot more polymorphisms in that gene. So, these are all clues. I'm not trying to convince you that this gene is the whole story, but it was very satisfying to find that even Kerry Stephenson, looking at his decode data, found that the QRS complex, which is part of the heartbeat, was radically affected in people who had myo-18b. Had myo-18b mutations. Now, here's the word of caution, because of course, myo-18b, this is the network it's in, right? So, okay, so we've hit one of these. Now what do you do? So of course, now we have to do proteomics, because obviously this gene has perturbed this network. But if you were looking for some kind of a causal effect, this might not be it. It might be one of these other ones. And so we're now stuck with the usual problem of polygenic scores, and what are we going to do about deciding where we might intervene to make a difference. But getting this far was a bit of a triumph for me, it felt like, anyway. Okay, here's one that's probably more interesting for George, which is COVID-19. All right, so what did we do? We found that indeed we were stuck there in March of 2020, and we were looking at this mess, which is diverse disease presentations, comorbidities, long-term effects, and we were being asked to provide a nice mouse model for this disease. And we were also aware of the fact that there was an enormous component of the host that was dictating how people responded to SARS-CoV-2 infections. And so, of course, we were trying to convince the world we needed mouse models of this, and obviously we did, because how are you going to do quick diagnostic therapeutics, antivirals, vaccines, etc., without them? Problem was that no mouse got SARS-CoV-2 because this Wuhan strain did not, the spike protein that normally interacts with the ACE2 receptor and gets into the cells in your heart, in your lungs, did not recognize the mouse ACE2 genes. So for SARS, for the actual SARS virus, a mouse had been made by another investigator that had a human ACE2 transgene being driven by an epithelial promoter. And these mice were terrific because you could use them for vaccine screening, but they were crap for looking at anything else because they died in 5 days, and that's not how most patients respond. So we turned to GeDi, and here's just a bit of a list for those of you who are interested in infectious disease. They've been used for influenza, they've been used for Ebola, West Nile, tuberculosis, and the COVID, SARS-CoV, in different studies. So we knew that these mice had the capacity to tell us something about it. So we decided to get over this problem that these mice had. These were C57 Black 6, by letting them go wild on our GeDi females. And so now what you end up with in the F1 generation is a bunch of mice that are genetically diverse, because half of their genome comes from one of these guys, and yet at the same time they have the transgene, so they can be infected. And so the question is, what happens? And the answer was everything. It was just great. Here's the C57 Black 6, and here we have, death immediate, like 4 to 6 days, they're all gone. As I said, we had sex bias. So if we pushed this one onto the NOD background, it was, the females were like impervious and the males kicked the bucket. Or the other way around, I'll show you a couple examples. Or if we went on to a PWK background, the mice walked away. And so we found that if we looked at this in great detail, we could find everything from resistance to sensitivity to sex bias in both directions, all just by putting that one mouse against these other 8 strains. So we felt pretty happy about that. And what we really liked was the parallel with the way these mice responded to the infection with what we were beginning to understand about how humans responded to the infection. What we found was the resistant mice had the strains that we crossed this ACE2 gene into that had resistant phenotypes had very high, very early interferon alpha expression, and the sensitive ones did not. And at the time, a woman named Iwasaki in Yale was looking at this in humans and discovered exactly the same thing, which was that interferon response that was very rapid and came down very quickly was synonymous with mild disease, whereas delayed interferon activation really let the virus get out of control, and that's what got you in the end. So we think that this actually was a pretty good model for the animal, and then went on, because we then, as the variants of concern came on board, we discovered that indeed you didn't have to have the transgene, because now the spike had mutated and they could recognize the mouse ACE2. And I raised a huge amount of fuss about this, because suddenly this is a virus that is essentially going to infect every mouse in every house. And so it was going to, I thought it would increase the capacity for this virus to get into the system and really wreak havoc. The good news is that, as those of you who are virologists know, viruses tend to get less extreme in terms of their infectivity as they mutate. And these viruses were mutating and therefore didn't cause as much trouble. But they still had a pretty interesting effect on Castaneus without any ACE2 from human, which was overexpressed. Here, we saw that the Castaneus genotype was very, very susceptible to the beta and gamma. And we went on and looked at all of them, and the same thing was true, whereas something like PWK was not. So the idea here is that there's going to be a capacity for each of these different these multiple phenotypes that you see in the mice, the multiple ways in which this virus affects humans, hopefully can be modeled. And we're actually doing an experiment right now with our friends at the National Institute of Health. I'll just go back for 1 minute, looking at the long-term effects of SARS-CoV-2 on the heart, which it turns out is well reflected in our transgenic, in our GeDi mice. So that's the kind of idea. You've just come in for the best picture. All right. Now, this mouse was not been photoshopped. This mouse actually had that expression with that gesture for the camera. And I just want to say, this guy, he has the little white stripe at the top, which means he's WSB, which means he's a wild-derived mouse. And this is the guy who will bite you harder than you can believe if you put your hand in. And he will, after he's bitten you, he'll jump out of the cage and run away. So when I send people WSB, I send knee pads with them, because they're going to be on their knees looking for them. Okay, one last story for those of you who are interested in life course, and then I can tell you my epilogue. So this was an experiment that I started a few years ago with Tony Wiscaray, who is a friend of mine out at Stanford, interested in lifespan, in particular the potential for plasma to tell us all about lifespan. And we got some money from the Altos group to do this experiment. The idea would be that we would take GeDi mice and run them through their entire life, no interaction, no intervention, no caloric restriction, nothing, just watch them live and die, and do enough of them so that we'd have statistical relevance, and do the things you can't do in humans. Take all their tissues at various stages, 1 month, 3 months, 6, etc., etc. Many of them are dead by 30 months. And then do all the omics, the plasma proteome, clinical chemistry, transcriptome. So we got this whole thing set up at JAX, and what was to use what Gary Churchill told us about. You take mouse A, in this case 10, number 10, and you made it to mouse B, which in this case is number 6. And then you take mouse B, 6, and you made it to mouse C, 3. And so each one is got a common parent, so that in the end you end up with a lot of combinations, but it's easier to do the sort of the deconvolution of what's going on genetically. And we looked at Black 6 as well. And of course, the lifespans were older than that, which were really exciting because there was huge differences. I mean, you know, twofold, threefold differences between these crosses. And I should say that these differences were not reflected in the parents. So you're looking at some kind of a hybrid vigor or hybrid emergence of some phenotype that otherwise you wouldn't be able to see. So to point out a few. These guys, these males, 3 times 4, they lived forever. In fact, they're still, some of them are still alive. And same is true of the females for 40 times 10. And again, I have to emphasize that this was not true of the parents. There are some losers here. 32 by 40, major losers. It turns out they all get vasculitis at year 1. I mean, they turn 1 year old and they all get small vessel vasculitis. Really great model, but it wasn't quite what we were looking for. And we looked at frailty, which is one of Gary Churchill's favorite ways of measuring age, and discovered that indeed there were obvious differences between males and females in this particular group. But what was most interesting to us was that if you have a frailty index, which is multiple different things that you measure in the mouse, we found that there were moments when all of a sudden there was a step change, so that it wasn't just a gradual disintegration, which we thought was quite interesting. And then also the timing was really different. The aging indices came on at different times. So if this is I'm just trying to give you a feel for this. This is these guys, 4119, and these guys here are 19 times 32. And you can see that they got the same problems, but they got them at slightly different times in their lifespan. So for us, that's quite interesting because these had more or less the same actual longevity, and yet things were coming up at slightly different times. So anyway, that gives you a feel for the sorts of ways you can set up these experiments, and none of these is really finished. I'm giving you just vignettes of things that we've done, and so I don't have the big nature paper at the end to boast about. However, one thing that I think is the future in this field. God, I'm not in the right place. I should be sitting here and talking to you like. Sorry, guys, I'm online. But what's next is, I think, to actually do collaborative cross-mark too. And the way we're going to do this is to go to wild mice. So the current GeDi mice, this is work of Beth Dumont, who's a brilliant geneticist at JAX. She measured the amount of genetic diversity in most of our strains, the ones that I showed you, compared to the wild-caught mice that she started to study. And it's humongously different. And in order to capture some of that genetic diversity, which we feel will give us lots more richness and hopefully more human relevant information, she teamed up with a friend of hers, Michael Nachman, who collected mice from different latitudes. And if you think about it, for wild animals, latitude is a huge evolutionary pressure because they have to live through either freezing cold or incredibly hot, dry, moderate. And each of these mice were domesticus mice, and they were caught by Nachman and his wife, who had to go around and capture them all. And then they started breeding them to get them inbred. And Beth brought them to JAX, and we derived them inbred. And now we have several of these, but she chose those 4 to start a new flock and sort of started to cross them up. And what she's finding in this wild mouse diversity platform is huge differences in their phenotype. Don't go into this, it's just a PCA plot, but the idea is they're much, much more different from each other at the phenotypic level. And they have about 45% more SNPs than any of the mice we have now. And here's just some data, very early days, but this is lean and fat mass. And that's interesting because the 2 that are from Edmonton and Saratoga Springs have higher fat mass and lower lean mass on average, sitting in the same mouse house, eating the same chow. So you know there's some genetics going on there. We're now outbreeding these 4 founders to make a new diversity outcross lines. So these guys are going to be hopefully breeding up very soon, and therefore we're going to hopefully be able to circulate them and get more information. They're all genomically characterized with T2Ts coming as well. So hopefully this will be a really good resource. Now, we're not finished, however. That's what's going on with the wild-derived inbred strains, the wild-derived outbred strains, but now comes the mouse genome project from wild mice, right from the field. You know, these mice are all wild animals, and so the idea would be that they will capture as much as possible, avoiding the inbred aspect of them. And furthermore, Beth, who's here, is the one that's spearheading this, and she's capturing information from mouse pelts in museums around the world. She actually, the domestic mouse actually originated in Iran, so she was all set to go to Iran to capture some mice, and guess what happened? She had to cancel her trip. Anyway, but the point being that we hope that these will capture wild mouse diversity and understanding the history of the wild mouse so that we can go back and understand possibly how these genotype to phenotype correlations got going, and then try to make some correlation between the lab and wild mice to contribute to pangenomic information. Here's just a little bit of data on these animals. We have about 154 wild mouse genomes, 36 inbred strain genomes, and a lot of really great SNPs to explore. And right now, you can find out more if you're interested, because we have a website, Wild Mouse Genomes Project, which I hope will be helpful to people who are interested in this kind of thing. Okay, just finally, before I quit, I just wanted to let you know about another hopefully helpful resource that we're developing. So people say to me, yeah, easy for you to say, you know, you have 500 mouse experiments running in concurrent oblivion, as if this was something anyone could do. And of course, that's not possible for a lot of places. And the UK is no exception, given the fact that I run a lab at Imperial and I can hardly get one mouse without having to sign 50 forms about what I'm going, how I'm going to pick it up out of the box. So what we've started to do is to consider making IPS lines out of all of these animals. And the idea there is that what we can hopefully not necessarily pick up in a particular derived differentiated cell, we could refer back to the mouse, which you can't do with human IPS in the same way. And so the idea would be, since everyone's looking at human IPS, we can make parallels between the human and the mouse IPS, but have the capacity to go back in vivo again afterwards. So that's where we're heading with not only these 8 founders, but with the derived mice that I discussed, the diversity across mice. We have about 200 lines. Well, why is that important? I just love this result. I'm going to just tell you this one result. So Martin Pera, who is the stem cell guru, worked with Laura Reinhold, one of our really incredible scientists who's interested in stem cells, looking at transcriptomic clustering by mouse strain in mouse embryonic stem cells. You'd expect that in development, I am a developmental biologist, I never thought in a million years there would be 5 different ways to get to Rome. But it turns out that you may get a mouse every time, but there are many routes to getting that mouse. And here you see an example of it, where we have 3 of the inbred strains from the founders, and yet they are getting to the first set of lineage commitment in many different ways. So that tells you that even within these highly inbred strains, there are going to be laws about how you put a mouse together that's going to be variable, which I just, yeah, I didn't believe it when I saw it. And what else can you do with them? The idea would be you engineer candidate mutations in GeDi mouse and human stem cells. Then you differentiate the stem cells into a cell type of interest. In this case, we were looking at neurons, because this is work of Martin Pera, who was trying to see if he couldn't study autism in a dish in a mouse. And then, of course, this produces the possibility for precision disease models that are in vitro. And this was a really beautiful study of autism in a dish. I highly recommend it if you want. This is they said it can't be done type of paper, but indeed you can pick it up. So what happens is in a person with autism, the neuron grows out and then it dies. And so that's why there are these lesions in the brains of children that essentially give them sort of gaps in their capacity to connect. And you can see it in a mouse, because the brains are smaller and they show signs of autism, and you can even see it in a dish. The neurons grow out and then they literally die. So it is crude. That's a crude one, but it's the beginning. Maybe there are ways in which we can use stem cells to at least complement, if not replace, animal work. So anyway, this is just one of these slides where I try to make the point that humans, we can get lots of data, but there are huge gaps because we can't cut them up and tell them who to marry and what to eat and how to get infected or not. And over here, the mice are much more, they're pliable. And so as a result, we can get more information. Now, some of that won't be useful because it won't relate to anything we see in the human, but there might be some things that actually do relate. And so we could fill in some of the gaps using the mouse. At least that's the theory. And ultimately, this is the sweet picture at the end to suggest that it's a good thing to have precision medicine at your fingertips. Anyway, lots of people to thank. I won't go into it. I've tried to mention people as we went along. That's the jacks. That's the ocean. It's really beautiful. I highly recommend that you come and visit us. And I'm thanking the mice as well. So that's the end of my talk. Thanks.
That's great. So people online can put questions in the chat, and I'll read them out.
Got a very boring question, which shows you how little I know about genetics of mice.
Yeah, you
If
can
we're
do.
crossing, say, 40 times 19, they're both homozygous.
That's correct.
So the children are always the same.
Obligate heterozygotes, which is just what you want because you don't want an inbred strain, but you want to be able to get to it, which you can't with these diversity outcross mice because they're single animals just like single humans. So taking two inbred strains and crossing them is the sort of the cheap ways to go. But I like it because at least we get models. I mean, we now have a fantastic model for premature leukemia. One of those crosses gave it to us.
Yeah.
And we can now dig in and ask how that happened.
Because the data chain crosses at the end.
Yes.
So 4x3 overlaps.
Yeah.
So they're both inbred.
Yes,
Parents. But you inbreed the offspring.
we do not. We just study the offspring.
So
because
they segregate out.
Yeah. So the point is that the minute that you cross the offspring, you're back to a mess.
I know, yeah.
Yeah.
But in your aging then, there must be
Yes.
huge variation. I mean, that must have been an average of the population who are all have different genotypes.
So there were 8 different genotypes only 8 different genotypes were developed because we took every one of those crosses was between 2 different inbred strains. The difference was with the daisy chain that it was as if the mummy and the daddy had kids. The daddy went off with another mummy and had kids.
But when you did the aging, you did 4x3. But to do the aging,
Right.
you've taken a group of offspring.
Oh, yes, but they were all genetically identical.
So you did inbreed the offspring.
No. They weren't inbred. They're obligate heterozygotes because if you have 2 homozygotes and you breed them
Yeah,
together.
there's lots of different meiosis there. So.
No, but the meiosis occur in the inbred, and who cares because they're congenic.
Okay, good.
No.
See what I mean?
Haplotypes are the same, doesn't matter.
Yeah, exactly. So in the parents, whatever happens, doesn't
You get like
matter.
one free meiosis without messing up sort of your.
You have one free meiosis.
Yeah.
That, of course, assumes that you're not going to see any mutations, which of course is ridiculous because you do. So this is of limited, it's not congenic in the same way.
Yeah, that's only in the first generation.
Yeah, if you kept going, it would.
But when you did the aging, is that off the F1?
All F1.
Well, let's see. I like that.
Yeah.
I did
So that
see
way you get.
40 was both in the least survived, like the
Yeah.
youngest time.
Notice that.
Yeah.
Isn't that interesting that
Like
one of
both.
the parents just, it's like if you have two wads, you're gonna get very different, the kids are gonna have different lifespans. I
Yeah.
guess that's obvious, but for us, it feels obvious. For the mice, it doesn't feel obvious.
No.
Yes.
And these are in cages? These are survival in cage or survival in group?
Oh, they are survival in group. They
Okay,
are survival in group
say
because mice
live
hate to be
longer.
alone. They live a lot longer when they're together.
right.
Yeah. They don't really like being alone. They're
Right.
miserable, actually. They die a lot sooner. This is all, mousy stuff.
Yeah, no, just,
Yeah. Anyway, sorry, Tom.
Pick up different effects if the mutations are on the father or on the mother.
Oh, I don't think we did, but I should have looked that up because somebody just asked me that question. I forgot to look it up. And of course, the person who asked me was my faculty who's interested in mitochondrial genetics. So, which makes a huge difference depending on who comes from where. So I have to go back and see if we did both ways. I suspect we didn't, which is stupid, but we only had so much money to do it. And it was already quite a few mice.
See, it's quite a lot of work.
Oh my God, when we had to harvest those mice, we would have like 4 graduate students sitting in a line, liver, spleen, yeah,
I've actually done that.
yeah.
Who's actually done the dissecting?
No.
So,
Yeah.
Is
this a question or you just want to mention it?
I did.
Maybe.
Michelle, I had actually done some proper work.
We have hippocampus. We have hippocampus coming out. Yeah,
Can we come to, presumably, and I'm talking maybe to the room people who know much more about statistical genetics than me, it's possible to kind of calculate G by G or some measure of
Yeah.
G by E just from that observed data that you get. Because if the 40 is associated with, very high lifespan, but also very low lifespan,
yes.
then you must know there's G by G. I'm hoping Michelle answers this as a sort of, statistical genetics person.
Yes, you could. But like, there's also some variation in housing that obviously might.
Yeah.
It's safety numbers at that point. You've got to integrate.
So
them.
I saw you have a couple of, you have like dozens per strain.
Yes, well, we had 6, we took down 6 males and 6 females every 3 months. So we have, sitting in my freezer are 80 boxes of bits of mouse. 80. It's just.
Yeah.
Yeah, it's a
So
lot.
there's like not quite a few per cross, basically.
That's right.
Yeah.
We did many per cross.
Right.
And
The
we,
lead lines aren't.
you have to because they do die.
Right.
Most of the time, they do die around 28 months or something like that. Yes.
Do you ever look at cross-fostering to see if the effects come
Ah,
through that?
that would be incredibly interesting. And we
Show
have
some
no,
stuff.
we should. I can send you your favorite one, the longevity one, the leukemia one, and we can cross foster them and see if it makes a difference. But I suspect, don't forget these are SPF mice, so they're not seeing a whole lot of activity in terms of infection. But
I have a question about the wild mice, which Mark I probably want to chip in about as well, which is, question is about, do you actually phenotype the original wild mice? Because obviously, there's a huge difference between wild mice and lab mice.
Yes.
And that's a mixture of the fact that the lab mice are all inbred, and even you've got genetic mice, and
yes,
etc. But there's that, and then the second thing is actually it's growing up and with pathogens. So obviously you bring in the wild mice, but then they're brought up in SPO, specific pathogen.
that's right.
Environment.
That's right.
So after by the 3rd generation, if you actually had phenotype data from the wild mice, you'd be able to make
Yes.
some statement about how much of the phenotypic difference is actually due to living in a live environment and how much is. Yeah.
Yes.
Are there any data on that?
I think there are some data on that. That would be the work of Michael Nachman, who collected these mice in the first place. And I highly recommend that if you have a shot at having Beth here, you would love, she would be able to talk your language to you. I am parroting what she's told me about those animals. And I think you would really enjoy the insights she has into microevolution and some of the other really interesting things that she sees. Just remember that even if you inbreed a strain coming from a wild mouse, you haven't put it through the bottlenecks of fecundity or docility
No.
yet. So you will have a much
Yeah.
broader spread of phenotypes from those animals than you would, even if they have gone through a number of generations. They still would not have been subjected to those inevitable things that happen if you're trying to produce these mice at scale, which is what we do. So that's where I think these mice will be interesting. Now, I have to say the Wild Mouse Genome Project is a genome project, and a lot of those mice are ex-mice. They're sitting as pelts in museums, so you're not going to get much phenotype out of that. However, you will be able to inform the whole way in which these mice evolved, which may tell you how the ones that you are dealing with are related to each other or not by evolution. So that, I think that's the theory here. And she's an evolutionary biologist rather than a translational biomedical. Anything from the audience? Any of you guys want to ask a question? Happy to answer.
Mark, that's the last question.
Well, I'll do it later, actually.
No.
It's just, I was thinking that was the work of Rosenthal, so that you might know.
Janet Rosenthal?
No, Stefan, but he was in the US. I can't remember the
Oh.
PI image. So they put wild mouse microbiomes into their mice,
Yes,
and then their whole immune biology changes
yes, of course.
fundamentally.
Yes,
And
yes.
this is generally relevant to all lab mouse work, but then on to genetic variation as well in the mouse.
So that's a great question, and we are still debating how to attack it. I made a big stink about 5 years into I've been there 10, so about halfway through. After 5 years, I said, are we not going to address the issue of microbiome at all? Are we going to be sending these mice around the world with different microbiomes? It's all very well to say that you're going back to the original genome, but what are you doing about the microbiome? So that set a flurry of activity through the production facility, and they, to their credit, did a careful study of microbiomes in the mice of various rooms in various centers. It all depends on what they're being fed. So basically, all the mice at the Bar Harbor campus were more or less identical in their microbiome. But if you went out to the Sacramento campus, where the mice are eating something slightly different, it was slightly different. So that suggests that you may send the mouse out to somebody, but within a very short period of time, that mouse is probably going to have a different microbiome. And as a result, the phenotypes are going to vary. There's no question about it, especially the immune system. So it's the elephant in the room.
Brilliant. Thanks again.
Thank you.